So greetings, everybody. I'd like to acknowledge that we're hosting this session on the unceded and ancestral territory of the Coast Salish peoples. That includes the territories of the Musqueam, the Squamish, and the Tsleil-Waututh nations, as well as the Métis chartered community of the Lower Mainland region. All of you across this province in Turtle Island are certainly aware of where you are and our importance of taking care of the earth, limiting the resources that we use unnecessarily, and being thankful for the land that we're on. So thank you for that. Next slide. So I'm going to actually ask Dr. Gill, who's the transplant academic lead for the division, to please introduce Dr. Andrew Adams, who is going to give us this talk. Over to you, John.
Good morning, everybody. Thanks, Adira. It's a great pleasure to welcome you all to this lecture. I know this is a topic of high interest. One of the real privileges of being involved in professional societies, both in Canada and the United States, is our ability to interact with folks who are doing cutting edge science and are at the forefront of innovation in our field. Through the AST, I've gotten to know Dr. Andrew Adams, who's our guest speaker today, quite well. Dr. Adams is currently Professor of Surgery and Chief of the Division of Transplant Medicine at the University of Minnesota, where he holds the John S. Najarian Chair in Clinical Transplant Medicine. He is also the Executive Medical Director of the University of Minnesota's transplant programs in solid organ transplantation. Andrew started his medical career first with an undergraduate degree at Brigham Young University. He then entered the MD PhD program at Emory University, where he graduated magna cum laude. He then went to do his surgical residency training at Harvard Medical School, MGH, and returned to Emory University, where he was a clinician scientist and really prolific, both in terms of his clinical work. He while he was supporting 3 R01 grants of over $12 million in innovative research, he was implanting 50 livers during the same time. So extraordinary accomplishments. I can only say he is one of the leading academic surgeons in the world. I'm very pleased that he's found some time to bring us up to speed on something that's all very exciting to us in xenotransplantation. And I'm just going to leave it to, I'm going to change his title to say, "Are pigs flying, Andrew?" So over to you, my friend.
Yeah, thank you, John. Thank you, Dr. Levin. Appreciate the invitation and the kind introduction. And it's I recently moved to the University of Minnesota about a year ago, and at the time, my wife wondered about the weather, and so now I have some friends that I can commiserate with for the winter. So I appreciate that. Let me see if I can get my screen shared here in the right orientation. Are you seeing my regular screen or the swap? Should I swap display?
Oh, perfect.
Okay, all right, great. Well, a few disclosures for research funding and consulting. But I just wanted to start out, this audience is probably no stranger to kind of the challenges that we face in transplantation. And as a surgeon, we try to distill that down to something fairly simple, but the two great challenges we face now in any organ transplantation is that we don't have enough organs. And then when we are able to get organs and do the transplant, those don't last long enough. And so those are really the two areas of focus of many people's research, including our own. And I'll talk a little bit today about our efforts in xenotransplantation. This is a well-worn slide that kind of just illustrates that point, in the sense that the number of people on the waiting list, for kidney or other organs, kidney in particular, is ever growing. And the number of donors, although we've made some strides with living donation and using expanding criteria donors, aren't nearly enough for the need that is out there. And I always like this slide that comes from HRSA's website, and it talks about, kind of, just to put it in more, I guess, practical terms, that every 8 to 10 minutes somebody's added to the national transplant waiting list here in the U.S. And as we think about the number of organs that are donated and the efforts that are have been put into place there, it's extraordinary the number of people who, with their generosity, can save so many lives, but yet also really telling how many people are still in need. just to kind of illustrate that point, if we look at waitlist additions, this is a paper that was more a commentary paper put out a couple of years ago by people that are involved in regenerative medicine and tissue preservation. But if we compare the number of waitlist additions here on the left to the number of people who die of end-stage organ disease around the world, it really, is 10 to 100 at times, depending on how you kind of calculate that. And then there's probably some number in between that would benefit from a transplant if we removed all the constraints regarding donor availability. And so waitlist additions, or the patients on the wait list that are waiting an organ, really underestimate the overall need when we look at that both, within North America and around the world. And so when we, in our research efforts in our group, think about how we can improve the field of transplantation and offer that to the greatest number of patients and derive the most benefit from that, not only are we thinking about increasing the supply of donor organs, but decreasing the demand by improving outcomes post-transplant, including optimizing medical management with novel immunosuppression approaches, et cetera. And some of that dovetails into the success that's been had more recently with xenotransplantation. But really, when we think about increasing the supply, and there are many different avenues of research there, some very exciting in the sense of looking at stem cell technology and growing organs. But really, I think even though we potentially could optimize living and deceased donation, the efforts there are really important, decreasing disincentives to donate, et cetera. Even if we're able to optimize that in the best way, we'd still fall short of the number of people that we could potentially treat. And so xenotransplantation, although the concept has been around for half a century, has really come to the forefront more recently. And I'll illustrate that with a few points here in the future slides. And so, of course, xenotransplantation is talking about using organs or tissues from one species and transplanting them into another. And there's always historical reports of this occurring, and some of it's more folklore than actual event. But the idea of using, one species tissues or blood products or organs has been around for at least since the mid-1600s. Now, of course, those weren't very successful. There's reports from a French doctor, Dr. Denis. I probably didn't say that correctly to the French Canadians listening in, but he was said to have tapped veins from farm animals and used for human transfusions. And the first patient of his was a teenage boy who miraculously survived that procedure, but it was probably likely due to the very small amount of blood that he actually received. And after a subsequent patients died from these practices, that was banned, and certainly his career was affected. So several reports, detailing other early use of organs from other species, but again, some of that is more folklore than actual events. But really, when we talk about clinical xenotransplantation, it was right around the birth of the field of transplantation for allotransplantation, human-to-human transplantation. And this is a report from Keith Reemstra, who was a transplant surgeon at the time at Tulane in the early 60s, and he was one of the first surgeons to use non-human primate organs for transplant. And he went on to be the chair of surgery at Columbia University and had other remarkable contributions in his career. But in speaking about this in the early 60s, of course, there were limited options for patients with end-stage renal disease. Dialysis was not refined enough and available enough for patients who needed it. And deceased donor kidney transplantation was in its infancy, and the outcomes were really poor, despite the reports, of course, from Boston and other places where they were doing allotransplants, some with identical twins with some success, others with high-dose radiation, steroids, et cetera, with limited success. This first report of clinical xenotransplantation was of a school teacher with renal failure, and where Dr. Reemstra used primate kidney, and did the transplant. And in a series of transplants, she survived for longer than 8 or 9 months, but additional ones really lasted between a week and 2 months, and all died of infections or rejections. And so this concept of using organs from another species for transplant has been around for the last 50 plus years. Additional heart and liver xenografts were also attempted. And Dr. Starzl, and this is more recently, if we fast forward as immunosuppression improved in the 80s and early 90s, they continued to have some efforts in this. And this is a report from the early 90s where Dr. Starzl used a baboon liver into a recipient with fulminant hepatitis B. And the recipient survived for over 2 months, indicating, the function and physiologic aspects of the liver from another species were able to support life, and ultimately died of an infection and stroke. And at the time, they noted that there was no evidence of antibody-mediated rejection. But just this concept of using animal organs for transplant, was still, at least in the scientific realm. And then probably for some of those who are on the call that are a little bit older, they can remember, and this was from the mid-1980s, a more famous case of a baboon heart into a child who was named Baby Faye by the lay press, who had hypoplastic left heart syndrome. And there, Dr. Bailey at Loma Linda did perform the surgery, and the heart functioned for a couple of weeks, and after about around day 20, succumbed to some antibody-mediated rejection, unfortunately. It was these cases that really drove the ethical debate surrounding the use of non-human primates, evolutionarily close to humans, and physiologically also probably similar. But that really, given their endangered species of chimpanzees and other old world primates, and then also the fear of zoonoses, as some of these more rare infections were, at least the concern was raised about transmission from one species to another, really led to a moratorium on clinical xenotransplantation. And so from the kind of around 1990 forward, there were no subsequent uses of animal organs into humans. And if we go back and look at maybe what some luminaries in the field of transplantation had to say about xenotransplantation at the time, some of them were more optimistic than others. And so, of course, Sir Peter Medawar, who was a Nobel Prize winner for his work on tolerance, his statement was, we should solve the problem of organ transplantation by using heterografts. In this case, a term used for xenografts at the time. And he thought that one day, if we tried hard enough, and maybe in less than 15 years, of course, this was much earlier, around his in the 50s and 60s, and proved not to be correct with the timeline. But he was quite optimistic given his knowledge of immunology and physiology. And Sir Roy Collin, who is a contemporary of Dr. Starzl from the UK, who was a pioneer in both immunosuppression and liver transplantation, his quote is, "Xenotransplantation is just around the corner, but it may be a very long corner." And I think that's proven to be the most correct of the statements. And in the last statement, which is probably most famous of the three, is Dr. Norman Shumway, the pioneer and father, thought to be the father of heart transplantation. And his quote was, "Xenotransplantation is the future of transplantation, and it always will be." And that's akin to John's comment about when pigs will actually fly. But I think, now with some of the more recent advents, and hopefully I'll convince you with the next few slides that probably Dr. Collin is the one who was probably most correct about this. It's been a long journey. I think the innovations that have occurred over the last few years have really placed xenotransplantation into the realistic realm of clinical medicine. And so that's why I entitled my talk today, "Have we finally rounded that corner?" And so when we think about donors for xenografts, I spoke, at least initially alluded to the concerns with the use of non-human primates, ethical concerns given their close proximity evolutionarily to humans, the potential infectious risk with zoonoses, their limited number and endangered status. But one advantage that they have is they're what we call concordant with regards to their biology or immunology, and I'll illustrate that in the next slide or two. More recently, over the last 20 to 30 years, pigs have emerged as probably the ideal donor for xenogenic organs. And in some ways, they counter some of the concerns that were had with non-human primates, in the sense that In general, they're more ethically acceptable, I should say, as they're used for a food source worldwide. They're easier to breed. They generate large litters, so the ability to generate large numbers of animals and subsequently organs for transplant. They generally have similar sized organs for adult humans, and the physiology is similar in some ways, but their immunology or biology is somewhat discordant. And so when we talk about concordant versus discordant xenografts, we're talking more about the expression of distinct carbohydrates on the cell surface, similar to what we see with the ABO antigens. These are thought to be, they're due to glycosyltransferases that during evolution have changed in the sense that pigs have certain glycosyltransferases and sugar moieties that decorate their cell surface. And over time in evolution, as we move to New World and Old World primates and on to humans, there were various enzymes that were lost through deletion and inactivation. And so one important carbohydrate that was initially discovered was one called alpha-gal, which was due to an enzyme. And this probably represents 70 to 80% of the antibody we have to pig antigens. And so, as I mentioned, most pig cells express this antigen, the sugar on the cell surface. It decorates numerous proteins and lipids on the cell surface, and it's produced by this glycosyltransferase. And over time, the expression was lost in humans and non-human primates due to a mutation. And so all humans and non-human primates to a certain degree have natural antibody or preformed antibody, as I mentioned, similar to the process that happens with the blood group antigens. And this is thought to develop through exposure to gut flora as in the early stages of life. So one of the first advances with regards to pig donors was the discovery of this antigen, and subsequently with the advent of cloning, Dolly the sheep, along with now in the late 90s and early 2000s, the generation of alpha-gal knockout pigs. And so this, along with the use of transgenic pigs, which expressed human complement regulatory proteins, really advanced the field from having a survival of pig organs in, say, non-human primates in a preclinical study from minutes to days. This was a really inefficient process compared to what we have today through the use of homologous recombination and breeding. It took years to generate some of these animals. And so it really wasn't the technology that we needed to make multiple gene edits. And so we knew from research in the preclinical realm that additional xenoantigens, other sugars, or other proteins that differ between pigs and humans would be necessary to be able to lower the barrier for rejection. Because most of these animals, or I would say that a wild type pig into a non-human primate and presumably into a human would be rejected similar to an ABO incompatible transplant where you see hyperacute rejection. And so really it was over the last 5 to 7 years with the advent of new technologies in genetic engineering, including CRISPR, which we're all familiar with, that has allowed us to delete, inactivate, and insert multiple genes at one time. And it's really this combination of genetic engineering and advances there, along with advances in immunosuppression, that have allowed a resurgence in xenotransplantation and the possibility to do clinical xenotransplantation. And this has been touted in the lay press over the past few years with headlines, such as this one, where we're going to end the human organ shortages through the use of genetically modified pigs. There's a whole host of headlines. One of the ones that I like the best is this one, where we're talking about pigs are good for more than just bacon. Many companies and a lot of support financially have been poured into this field. There are 4 to 5 major companies now, biotech companies that are startups, including this one that is a subsidiary of United Therapeutics that was at the center of some of the more recent clinical xenotransplant, called Revivicor. This is their conceptual illustration of their pig donor facility where you can produce thousands of organs for transplant each year. They have little landing pads for helicopters to come in and take the organs, or drones now, and take the organs away. And so you can see that over a number of years, this has evolved, but and used to be conceptually more scientific or more science fiction in nature, but now I think is closer to reality than some of us had imagined. Another company that's been has received a heavy amount of funding is eGenesis, founded by George Church, a geneticist out of Harvard, who's looking at using multi-gene, where they have a report in Nature of editing out 60 plus genes at one time in one pig. And it's interesting that some of the major financing efforts are led by Fresenius, DaVita, and Baxter, as they see where the field is going. So where are we now? I'd like to just spend 5 or 10 minutes, sharing some of our work that relates to what we've been doing in the preclinical realm as we strive to figure out what's the right, genetically modified pig, what immunosuppression should we be using to be successful. And we do that in a preclinical model using primate recipients. So as close as we can get to the human scenario, and then have genetically modified pigs. And I'll give an update, our work in kidney. There are others who've worked in heart, lung, and liver. I'd say for kidney, if we look over time, this is a review paper, at least a figure from that review paper entitled Bringing Home the Bacon, from David Cooper, who's now back at Harvard. But looking at the survival in days of a pig kidney in a non-human primate recipient. You can see in the early 90s, there's really not much success with the advent of the gal knockout pig that I mentioned, and some transgenic pigs that expressed human complement regulatory proteins like decay accelerating factor or others that can help control the complement mediated cytotoxicity that occurs when antibody binds to the endothelium in an accelerated antibody mediated rejection. You can see some increases in survival up to almost 3 months. And it's really more recently, and here's where our group got involved in kind of the mid 2015 range, where we started to couple some ideas about clinical kidney transplantation and novel immunosuppression, where now we have animals surviving out over 3 years. So just supporting the notion that a pig kidney physiologically can do all of the things that a human kidney can do, including, making enough erythropoietin for hematopoiesis, acid-base balance, calcium phosphate regulation, et cetera. And so when we think about rejection, when we entered the field, I mentioned hyperacute rejection, of course, that we're all familiar with. In the case of xenotransplantation, we have antibodies that bind to pig glycans on the cell surface on the vascular endothelium, and it proceeds much like hyperacute rejection. And then as we knocked out the major antigen in gal, we get to a point where we see accelerated antibody-mediated rejection. And these are due to antibodies directed at other antigens other than gal, but it's similar to an early antibody-mediated rejection, what you would think. And so here again, some of the strategies to counteract that, we delete the antigens that aren't found in humans in the pig, or in non-human primates in the pig, to make them more non-human primate or human-like. And then we add complement regulatory proteins that would control both the complement and coagulation cascades to prevent those from taking hold and causing destruction of the graft. So as I mentioned, when we started, the median survival time for pig to non-human primate kidney transplant was around a month. There was isolated reports up to about 3 months, but no one with really long-term survival. And so our hypothesis was really that the driver of that early rejection in the first 100 days or so was directed at non-gal antigens, and that may preclude long-term renal allograft survival. And so if we treated the non-human primate similar to the way we treat patients, in the sense that we do a cross match at the beginning, much like ABO antigens, when you look especially in neonates, there's a bell-shaped curve of the titers of anti-ABO antigen or antibodies. Same thing with anti-pig antibodies. So we went into our colony to assess this. And so this was conceptually what we were thinking about. Primates or humans have antibodies directed at these antigens on the pig cell surface. So here's the pig with a lot of gal and some other glycans or sugars decorated on proteins that are not gal, but are different than what we find in humans or non-human primates. And so we have a variety of antibodies, the major one being gal. And so what would normally happen, these antibodies would bind to the endothelium, you get complement activation and subsequent destruction of the graft. Oops, went too fast there. And so what were some of the strategies to counteract that? Of course, the gal knockout, where we get rid of those antigens. What persists, though, are a smaller number of non-gal directed or non-gal antigens to which we have antibodies. And then we can add in things like a transgenic donor for human decay accelerating factor that prevents the formation of the membrane attack complex in the complement cascade. So if we still have high titers of non-gal antibodies, we can still have destruction of the graft. And that's what we thought was occurring at the time or hypothesized. But what we found out is if you could select animals that had lower titers of antibody to these non-gal antigens, and so we did that by screening the colony, just like you would a cross match and pick the right recipient for a kidney transplant, we could avoid that. And so here's the data for that. This is essentially a flow kind of old school cross match where we take cells from the pig, serum from a variety of different animals, 30 or 40 animals here, and looked at their titers over time. And what you could see is some animals had very high titers of anti-pig antibody titer and some had very low. And so we said, well, let's just select the 3 or 4 animals that have low titers and compare it to a high titer and see if there's any difference in survival. And so we developed this model where we take a non-human primate, in this case a rhesus macaque, and nephrectomize it. So the kidney that's working in the animal is the pig kidney that we implant. And then when we reperfuse and watch the animal over time for survival. And what we found, interestingly, was that not unexpectedly, when you select out animals that have low titers of antibody, you don't see early antibody-mediated rejection. You can get long-term survival out over a year. And this was the first time that that was reported versus a high titer animal where we see early antibody-mediated rejection, much like you'd see for accelerated, not hyperacute, but close to hyperacute rejection antibody. And so when we started, the reported literature was, again, like I mentioned, 2 to 3 months. And then on our first series of transplants using this selection criteria, we were able to extend survival out over a year. And then we looked, it allows us to examine kind of what's happening in the graft. Interestingly, when we go and take the kidney out, we can look at what T cells, for example, are infiltrating the graft. Do they participate in rejection? And are we able to target those with novel immunosuppression? And in an allograft, when we treat with certain immunosuppressants, it's a predominant CD8 mediated rejection. Interestingly, and this has been known previously, in xenografts, CD4 T cells are more important. So we were able to do studies where we could look in vitro, doing simple MLRs where we take pig cells, combine them with non-human primate T cells and look for proliferation, and showed that the CD4 T cells proliferated more efficiently than CD8 T cells. And then we could separate that out in the model that we had and said, well, let's just get rid of, by depleting with an antibody, CD4 T cells or CD8 T cells in the primate to really understand what's the biology behind the rejection. All of the animals in this study had low titers of anti-pig antibody to begin with. But what we saw is a very striking difference between the animals that had their CD4 T cells depleted, where they had prolonged survival, versus those that had CD8 T cells. Again, kind of insinuating that CD4 T cells play a principal role in early xenograft rejection and probably drive antibody production, much like we already know the biology, but just proving it here in a preclinical xenotransplant model. And then we looked at antibody production over time, and some of the new medicines that we're using as immunosuppression targeting co-stimulatory pathways do a very good job at preventing the development of new antibody over time. So to date, if you look at all long-term survivors, regardless of the organ type in the preclinical models, meaning pig to non-human primate, kidney, heart, islet, others, they've all relied on some type of therapy to block the CD40, CD154 pathway. If we use things like tacrolimus, we have very limited survival. So every variable is the same. the mainstay immunosuppression is tacrolimus versus, say, anti-CD154, an antibody to block this T-cell co-stimulatory pathway, we see survival times in a month to 2 months versus a year. And so it's a key component to the success. And this is true of the most recent human clinical heart xenotransplant, is that they used an antibody against CD40. And so the development of these immunosuppressants is going to be key to the success of clinical xenotransplantation. And so this was true not just for our group. A group at UAB showed a similar result. If you looked at using antibodies to CD40 in this case, or tacrolimus, significantly prolonged survival in the group that used these novel antibodies for immunosuppression. And so what is the importance of this pathway? We know it's important for T-cell activation, for TB interactions, and it's being developed for kidney transplantation in a, I guess, redeveloped, I should say. These antibodies were on the forefront in the lay press, in clinical trials in the late 90s, and unfortunately, because of higher incidence of thromboembolism and rejection, their development was halted. But they've really found a resurgence in the last couple of years, particularly with the re-engineering of those antibodies to prevent platelet adhesion that led to thromboembolism. And so, and there are multiple clinical trials evaluating some of these. They still have some issues, but I think we're on the cusp of having some of those close to approval. But what will be key is their use in xenotransplantation. And here we were evaluating, do you need to block the ligand, CD40 ligand, or CD154, CD40, the receptor, or tacrolimus? And what we found, even though all of these had similar levels of anti-pig antibody to begin with, really striking difference in survival. As I mentioned, the animals treated with standard immunosuppression involving tacrolimus and some T-cell depletion up front, MMF and steroids, were the worst and only survived maybe a month or so at the longest, and a median survival of only 12 days. The anti-CD40, the targeting the receptor, and it really depends on what antibody you use, was significantly better, but still inferior to targeting the ligand. And so when we looked at these, you saw early acute antibody-mediated rejection, some evidence of thrombotic microangiopathy that could also be due to antibody injury, and dysregulation of the coagulation cascade. And versus the long-term survivors, where we start to look at the development of anti-pig antibody over time, where some of them develop high levels of antibody but continued to function as we look at that. And we saw some more chronic changes, which would be akin to some allograft nephropathy, in this case, some chronic glomerulopathy and an interstitial fibrosis. And so developing genetic modifications to the pig to prevent this, or additional changes to immunosuppression to prevent the long-term failure of these xenografts is now some of the focus of our research. So again, why did targeting the receptor make a difference versus the ligand? If you just block that interaction, why would they not work equally the same? And this line of research was prompted in part by some of the work by a coworker, colleague at Emory, Dr. Mandy Ford, who did some work in allotransplantation. And here she was looking at the differences between anti-CD40 and anti-CD154. And this is in a mouse model of skin transplantation, but to elucidate some of the biology, is there another receptor, for example, for CD154? And that's what her conclusion was, that CD11B, a well-known marker on macrophages involved in innate immunity, an adhesion molecule, can also interact with the ligand for CD40 to probably cause some of the findings that we found as well. And so she found that if you treated with anti-CD154, it was better than anti-CD40, similar to what we had seen in the xenograft model. And when you now combined it with an inhibitor of anti-CD11B, they could restore the function to similar to anti-CD154. And so we took the same tactic. We compared our groups that were treated with the antibody against the ligand versus the receptor, or a combination with this new target anti-CD11B, and showed that these are ongoing experiments, showed that they were similar to when you block the CD11B. And so that might be that interaction between the adaptive and innate immune response through CD154 that may predispose to early rejection when you're just targeting the receptor. And so just conclusions from our studies in the non-human primates is that really the key to preventing early antibody-mediated rejection or treatment with antibodies that target the CD40, CD154 pathway. And in the past, people said, well, those aren't clinically applicable anymore because they failed in clinical development. But now we see that there are upwards of 6 different antibodies to either CD40 or CD154 that are in some stages of clinical development, including Novartis's iscalimab, that had some challenges in the kidney transplant clinical trial in patients, is continuing in liver transplant. But I'll show you some of the work we did with that antibody as well. What about B cells? Antibodies important. This model allows us to deplete B cells with an anti-CD20 similar to Rituxan, and say other groups have shown in a non-human primate cardiac transplant model that this is important. And so we wanted to look if we combined our regimen where we were getting rid of CD4 T cells. This is an antibody dosed at the time of transplant. They reconstitute about 6 to 8 weeks afterwards, and really don't show many signs of over-immunosuppression or infectious complications. But here we wanted to say, well, what happens if we deplete B cells at the same time too? And there wasn't a big difference, surprisingly, between just CD4 depletion, suggesting that maybe the T cells are really driving the B cell response. But we have one animal that's now close to 3 years that shows that that regimen and that type of kidney can support long-term survival in a non-human primate model. And we went on to look at the phenotype of the B cell there, and suggested that there may be some advantage, and many of the clinical regimens that are being proposed now involve early depletion of B cells along with T cell depletion and complement inactivation. And so this is just work from a couple of other groups to inform you kind of where the field is. This is a group from MGH using the kidneys that we used. Also subsequent other pigs that have multiple gene edits, upwards of 10 edits, to show that they're improving survival over time. And it can show you the different antigens that are knocked out. These are additional sugar moieties or glycans that have been knocked out in the pig to make them more human-like, complement regulatory proteins that are inserted as transgenes, and then other things that would impact the immune response to a xenograft, things like human leukocyte antigen E involved in the NK cell regulation, CD47, another marker on cell surfaces to prevent phagocytosis from macrophages, and PD-L1, which helps to inactivate T cells when it binds to PD-1 on unexhausted or activated T cells. And just to show you that they also have started to see longer-term survival when using the technique that we published on selecting animals that had low titers of antibody. They did see some infectious complications. Their regimen's a little bit more rigorous than what we had used. Lung transplantation, there's been advances, but those advances are much, I'd say, compacted in time. So before lung transplantation, as you might imagine, there's a vast amount of endothelium. It's a more challenging model. And so what used to be a few hours, this is now survival in days, not years like we talked about in the kidney model, where we see survival of lung transplants, pig lungs into non-human primates, going from a few days now out to up to a couple of months in selected animals that have multi-gene edits. And then this is just showing you again the number of gene edits that are starting to emerge in some of these pigs, multiple gene edits. The challenge has been that scientifically you'd like to change one variable at a time and see if it makes a difference and understand the biology that's affected by that. But that's not necessarily how the field has chosen to proceed. These are multiple changes in new models, and it becomes hard to interpret, are these really necessary to proceed what's the role of intellectual property that's driving the companies to make some of these edits, et cetera. And so lastly, just looking at liver transplantation, much akin to lung transplantation, areas where there'd be great need for xenotransplantation, but limitations in the survival. And so here, again, what used to be a few days has now gone out to a month, but the longest surviving animal here in the pig to non-human primate liver transplant model is about 4 weeks. And then lastly, this is the pioneering work done at the University of Maryland, where they were doing pig hearts into baboons, initially heterotopically, meaning into the belly, where they saw survival times of out to 2 or 3 years. And this really formed the foundation for their work that led to the pioneering transplant that occurred, a month or so ago that I'll speak about in a slide or two. What really advanced the field in xeno heart transplantation more recently was the group in Germany, where they started to look at additional genetic modifications, including the growth hormone receptor to prevent growth of the heart. There's still some debate as to whether the growth of the heart they saw in some of their studies was due to intrinsic pig growth or related to changes in blood pressure or rejection. Regardless, the pigs that they started to use included multiple gene edits, one of which was the growth hormone receptor. And then they also started to use ex vivo perfusion to allow some period of time between when the organ was removed from the pig and when it was transplanted, and allowed early for orthotopic heart transplantation, better early function. What was the problem before is they put the pig heart into the baboon, there'd be dysfunction and there was no way to kind of support the animal through that period of time. And so this similar strategy was also used in the clinical xenotransplant that was performed. So just to highlight a couple of those exciting events that occurred over the last 6 months, the first report came out in the lay press from Dr. Bob Montgomery's work at NYU, where they kind of developed a recently decedent model, in the sense that brain dead donor was then the family was consented to be able to put pig kidneys into the brain dead donor and watch for a number of hours as to the function. And so this happened last fall and was reported on. This is Dr. Montgomery and his team. Here they put the pig kidney and attached it to the vessels in the groin and had it exteriorized. And for a period of a day and a half or so, we're able to watch the function over time and take some biopsies and see what happened as a foray into clinical xenotransplantation again. So certainly a step forward, a significant step forward, and we're starting to learn more and more about the biology there. Again, this is the pig kidney that's attached, perfused, and starting to make urine. More recently, we learned of the report from the University of Maryland, and this has been in the press a lot, and I think most of you are probably aware of a recipient who was ineligible for a allotransplant, a human heart transplant, that had significant heart failure, arrhythmias, et cetera, wasn't a candidate for a destination ventricular assist device, and was approached under a compassionate use allowance to get a pig heart. So in this sense, this is a genetically modified pig heart that they had on the pump that was kept in ex vivo perfusion while they removed his heart and then replaced it. This is Dr. Mohammed Moeeden, who's the researcher that really pioneered this work at the University of Maryland, and Dr. Bart Griffith, who's the surgeon who's head of their heart and lung transplant group. And here's Dr. Moeeden, the patient, and his son. And so we continue to get sporadic reports now out over a month with a pig heart implanted, starting to do rehab off of ECMO, with the pig heart continuing to function well with no signs of rejection. So really an outstanding result, and I think the dawn of a new era for clinical xenotransplantation. Certainly some ethical, I would say, concerns as some of the details have come out, but I think they've done a great job of trying to balance, caring for the patient, the risk associated with this, and the bioethics that came into that case as well. The last report kind of for clinical xenotransplant that just came out in the literature recently was a report from UAB, where Dr. Locke and her colleagues approached another brain dead donor's family for permission to put in pig kidneys. And so here they used genetically modified pig kidneys, removed his kidneys and replaced those with 2 of the pig kidneys and evaluated those over time. Again, this is the pig kidney in situ in the abdomen, and we're able to report on that. And, unclear what some of the results can mean from that, but certainly we're starting to learn what's the biology, immunology, does the preclinical model in the primate replicate this? I'd say in the 2 kidney cases, standard immunosuppression was used, tacrolimus, et cetera, and short-term follow-up as in the decedent model with only a few hours, kind of 50 to 60 hours of follow-up, versus the heart transplant model where a novel anti-CD40 antibody was used. And now we have up over a month worth of survival there. And so again, I think it gives us a little bit more information about the importance of these novel pathways to the success of xenotransplantation. So what is the ideal pig donor? We start to think about all the genetic modifications that we could make, but which ones should we make and which ones are relevant? And certainly there are differences between primates and humans, and so our model can't replicate everything, but it does a good job of telling us what's important and what's not. And so, as I mentioned, we need to have something that's going to be used clinically for immunosuppression. And all of the data that we have in the primate and some of the emerging data that we have from now these few cases into humans suggests that the CD40-154 pathway is critical. And in the past, as I mentioned, the criticism was, well, this isn't a clinically relevant, pathway to use. But now, there's at least 6 different antibodies that are in clinical development or in clinical trials. And so I'm sure one or more of these will emerge both for allotransplant and for xenotransplant in the next couple of years. The glycans, I think most have settled on that these sugar moieties are the most important, but there may be other antigens that need to be deleted in the pig to prevent early antibody-mediated rejection. We need to potentially regulate the complement pathway, and that can be either with the insertion of transgenes, which can be problematic because the more changes you make in the DNA, the more potential you have to have off-target effects, tumorigenesis, et cetera. And so pharmacologically, we can control the complement cascade, and we've done some work using an anti-C5 antibody similar to eculizumab. This is a Novartis antibody that can control the complement cascade in the early post-transplant period. And it looks like that's for the first couple of months is all you need to do to get long-term survival, at least in the primate model. But there are other regulatory proteins for the coagulation cascade, thrombomodulin, et cetera, that may be important. These may be different depending on the organ that's going to be transplanted. The lung may be very different than the kidney, than the heart. And then other immune regulated or physiologic important genes like the growth hormone receptor for a heart, where we haven't seen that problem in kidneys with the growth. Or do you use a miniature pig model, miniature swine model? How many genetic modifications are too many? What's required for success? What is the FDA going to allow? What data are they going to need for approval? Those are all questions that several groups are grappling with as they move through the regulatory process right now. So what are the remaining questions? Well, I think the preclinical data that we generate in the primate is key. We have to ethically, especially in kidney transplantation, where we have dialysis and we have allotransplantation, be able to convince ourselves that what we're doing is right ethically, it's safe, and that we have some expectation of efficacy. And what is that balanced out with regards to the opportunities that a patient with end-stage renal disease might be offered? Again, that relates to which patient population is it ethical to enroll in a trial to start until we know what the success will be and the complications might be. And then what's the expected benchmark for success in this trial population? What do we have to have a reasonable expectation will occur to be able to proceed with the trial? And, we and others, John and I are on a panel with the National Kidney Foundation to try and address some of these questions and start to think about it as various groups construct their trials. We want to do it in a way that's ethically sound and reasonable and would be what we would want if we were a patient. So to summarize, significant progress has been made in xenotransplantation. These preclinical studies are key to be able to have the data support the initial forays into clinical xenotransplantation. There are concerns about safety and efficacy that we'll be able to address. But I think when we start to think about that, we have to consider what the outcomes are going to be. These are my collaborators at the University of Minnesota, at Emory, and at University of Miami. Dr. Techner and I have a few grants together to be able to support this work. And then lastly, I hope we're not, somewhere on this type of road where we only round one bend to go to the next. And I'm sure, hopefully we're more going the last curve and coming around that corner to where we can offer, unlimited supply of organs that would generate a similar expectation for survival for patients over time. So thanks again for the opportunity to share this forum with you and present some of our work and look forward to the questions and discussions.
Andrew, thanks so much. Really a tremendous amount of information encapsulated, and I think it gives people a really realistic understanding of where we are right now. I want to just, I've opened the chat up to anybody to please post a question in the chat. Or please raise your hand. I'll ask Adira to help me negotiate that because there's lots of people online. Andrew, I'm just going to start with one question, and you alluded to this. Our ability to splice in or splice out genes almost exceeds our ability to understand the effects of those individual changes. And so these development of these cassettes, which, as you said, might be a bit of a proprietary kind of thing. And so my question is twofold. Number one, we know that one of the concerns in the non-human primate models is animal welfare. And so taking out a whole bunch of genes, although it seems attractive, may have impacts that we even see in the non-human primate models that are unintended. And the related component of that is we all know the reality is someone's going to have this secret gene editing sequence then in their proprietary cassette, and the need for transparency around this which could really impede the progress that is to be had. So your thoughts? It's kind of a two-level question. One, a scientific one about that balance, but the other one, which is more of a transparency and a need for us to really maintain confidence of the public as this innovation happens. So over to you.
Right. No, those are both key questions that the field is trying to grapple with right now. And I think, for the first one, ideally, you'd like to make a single gene edit, test it, have your experiment, see what the differences are. And, there are various constraints on that. The number of animals that we use, as you mentioned, for animal welfare concerns, et cetera. But it's almost like we, in some instances, have gone significantly further without understanding kind of the implications. The issue with deletion of genes is a little bit simpler than the insertion of genes. And so deletion of genes, you can prove that it's been inactivated and that's not able to be transcribed. And so the gene is absent. You can look at you can characterize that in the pig. The transgenic expression of genes, as you're talking about some of these cassettes that land in 4, 5, 6 genes in one locus, and understanding how they're expressed and the expression pattern, not just in one tissue, but in multiple tissues and organs, is going to be critical, especially for the regulatory agency. And from one pig to the next, are they equally expressed in all the tissues? Where are they found? Where do you need them to be? And then do they land in the spot where we have certain safe harbor zones where they know they can land in cassettes of genes and it won't, at least from the data that we have, it won't have significant implications, but nobody has enough data right now to know that. And so, if you happen to land in a lot of transgenes in the past were just randomly inserted, and you hope that you got a location that was well expressed, but it could also land somewhere in a gene that's important for development, for tumor suppression, et cetera. And so those are the other concern on some of the transgenes is the expression of proteins that might serve as viral entry proteins. And so the potential, and we know some of these actually serve as viral entry proteins. And so that's the concern, I think, as we make these multi-gene edits and as you mentioned, to transparency, because as we start to think PERV, for example, porcine endogenous retrovirus was a key part of the studies that started to emerge in the late 90s, early 2000s of what kind of stopped the xenotransplant effort, then the fear of zoonoses. And what turns out clinically, there's been multiple instances where pig tissues have been inserted and transplanted, and there hasn't been any concern with transmission, but groups have now inactivated all of the different PERV elements within the pig genome, but that's 60, 70 different gene edits. And what is the effect of that? And is that more driving for proprietary concerns versus actual biology?
Yeah, the next question I think was partially answered there. It was related to zoonoses and really just asking you on specific strategies. So Andrew, do you see the gene editing approaches to elimination of PERVs as the main thing? There's obviously the production facility constraints and so forth, but do you see the need for let's say post-transplant therapies, antiviral therapies, et cetera, in that realm? So just a comment about the zoonoses.
That's good. I think that's the other Dr. Gill who asked that question. So good to see you, Justin. I think, yeah, it's a critical concern that I think with the data that we've accumulated over the last 20 years is maybe PERV is not as much of a problem as what it once was thought to be. And so there are certain subtypes of PERV that if you avoid, you're likely to have, if you did have transmission, you're likely to have something that you could control with common antiviral reagents that have been shown to be effective, at least in vitro. The clinical experience with dozens of patients now suggests that that's probably not a problem. That being said, as we use novel immunosuppression and inhibit different pathways, there's the possibility that might be something. So some of the suggestion regulations by the FDA, talk about having to store samples from not just patients but providers for long periods of time in case this comes up, and to be able to go back and study that and come up with strategies to do that. As you mentioned, the containment facilities, the biosafety regulations that go along with that are all aimed at being able to minimize that possibility. It's not zero, but I think it's low enough that most people feel like zoonotic transmission from these highly regulated pigs that are bred and born via C-section in a biocontainment facility and tested for multiple different types of viruses and bacteria, et cetera, the risk of that being a problem is pretty low.
Great answer. Thanks. Go ahead, Adira.
Just one, yeah. You talked about the work that you and John are National Kidney Foundation. I imagine patients are at the table sooner than later, and it's really interesting to see, because managing the message and managing the acceptance of this, so the reaction from patients.
That's a great question, and something that John has really championed and pushed for throughout his career, of course, and in our work right now. And so having their involvement, and that's one advantage to partnering with the National Kidney Foundation, is it's such a strong group to begin with. But it's that's to get their perspective. Somebody that's looking at facing dialysis 5, 6, 7, 8 years, and the mortality rate and the morbidity that's associated with that. What are they willing to accept versus we as clinicians or scientists, and what we assume that they're willing to do, and those are all the questions I think that we'd like to start to at least have a discussion about, because there's dialysis is great, in the sense that it's really when there was no potential solution for end-stage renal disease, but you and your colleagues and everybody else knows how morbid of a therapy it is, and how many people, depending on what your other comorbidities are, die on dialysis each year. And so, that's when you start to think about what are the trade-offs, what are the risks and benefits, what is a patient willing to do, what ethically is the right thing to do, and what are we willing to do as clinicians.
And patients entering into clinical trials, it's interesting, patients will take different risks in clinical trials than regulatory will let them. And I think that perspective is hugely informative when you're designing these trials.
I just wanted to put in a plug, Andrew, in Canada, Adira and others have really worked hard to assemble patient groups who can really inform these questions. So I really hope that there's an opportunity for, patient voice broadly in this, and I think that's really, really important.
Yeah.
I wanted to ask you one related question, which is, you highlighted the fact that some of the therapeutics that are really being pulled forward in the xeno space haven't really been tried and true in human trials.
Right.
And the interesting thing that's happening here is the innovations that we're seeing from the xeno side are actually propelling sort of stalled innovation on, the anti-CD40 ligand discovery. So there may be untoward benefits as we think on Xeno for allografts. And I just want to get your comments on how far do you think we need to have those trials spelt out in humans before you think it's holding up the progress in Xeno?
Yeah, absolutely. That's a huge consideration, because, typically you think about, pharma wants to develop, X new antibody and they look at the space. Well, it's autoimmunity. There's transplant. Let's do a clinical trial. And 7 years later, we get approval for, or may or may not get approval for a drug. And then in the middle of that, if somebody comes and says, hey, I'd like to use your, medication that you've had in development and have spent, millions of dollars on developing in a xenotransplant trial, and trying to convince them that that's the right thing to do, trying to convince regulators to not penalize them if they see some untoward effects in a high-risk trial like for xenotransplant. But absolutely. And I think the tide is turning, and some of these, I'd say some of the real benefit of these recent reports of clinical xenotransplantation are that people, the lay community, along with the pharma community, are saying, well, this maybe isn't as far-fetched as what we once thought it was. And maybe we should actually consider, using these reagents in clinical trials for Xeno, because the upside, if successful, is so high. And so I think that's really started to turn the conversation in many boardrooms now, suggesting that. And that small company that used the anti-CD40, that antibody was developed in the preclinical realm, is developed for use in the non-human primate. And it was going to be developed in autoimmunity. And now they've, through a compassionate use, request, used it in this heart xenotransplant model. And so I think your question is right. I think now these cases are really going to propel the development of these, which will be, if successful, advantageous for allotransplantation too. I mean, all of the data we have in the preclinical realm suggested those pathways are much better than something like Bella. And so we know what the long-term benefits of belotasib are, to be off CNI long-term, et cetera. But we know all the baggage that comes with it in the early term, and that's why nobody uses it. But if we had something more effective, or had a strategy that's more effective, that we can employ this to a CNI-free regimen for kidney transplant, where we can increase survival, decrease morbidity and complications. Yeah, I think it's going to be beneficial to not just xenotransplantation, but allotransplantation too.
So, Andrew, we're unfortunately at the end of our time. I lament that we don't have more time, but on behalf of everybody here, I'm sure we will all join me in thanking you for your time. I just wanted to close with one thing. Andrew is the Nigerian chair, and I just want to highlight that in May, the University of Minnesota is holding a special event in celebration of Dr. Nigerian's contributions to the field of transplantation, which are vast, and only to say that this is a rapidly moving field. Progress happens in fits and spurts, and I really welcome, I'm really grateful for Andrew's willingness to participate here. This is an exciting time, and I look forward, Andrew, to many more interactions. Thank you very much. I appreciate it.
Thank you, John. Thanks everyone for tuning in.
Thanks,
Thank
dear.
you. Thank
Bye-bye.
you.